I'm Shannon Salcido, and welcome to my presentation on DNA transcription and translation. This is for Biology 1011 for the summer of 2025. I don't know about you, but these were two words that had a very different meaning to me before I did some research, and I'd love to share with you what I've learned. Before we do anything, I need you to kind of flip on your imagination here and imagine that good old Miss Frizzle with the magic school bus showed up today, shrunk you down to the size of a cell. That's the only way we're going to be able to learn anything about the processes of transcription and translation. Let's go. First, we're going to talk about what we already know about DNA, the what, the why, the how. That's going to lead us into our discussion of RNA today. So we already know that DNA stores our genetic info. That includes the instruction for building and operating all living organisms. We know that DNA has a double helices shape, which is going to be important in our processes today. It's also important to the DNA function. DNA contain 4 different bases. We're going to need to remember these later on. We have adenine that pairs with guanine, and cytosine that pairs with thymine. Interestingly enough, humans are remarkably similar. Most of us share about 99% similar DNA. So even though we may look different, we're not really that different. So how is RNA formed from DNA? Well, firstly, RNA is a molecule similar to DNA that plays a critical role in protein synthesis. In other words, how cells create protein, as well as gene mutation. We know that protein is necessary for structure, function, as well as metabolism. Therefore, the creation of RNA really is critical for life. So what is transcription? It turns out transcription is the process of creating an RNA copy of an existing DNA sequence. So let's look at that. Transcription's primary function is to create messenger RNA. From here on out will be called mRNA from a DNA template. You have existing DNA, mRNA comes in, pairs with those bases, and creates a new DNA mRNA strand. This mostly occurs in the nucleus of eukaryotic cells, and it does include 3 distinct processes: initiation, elongation, and termination. And to understand this, we're going to take a quick look at all 3. So, initiation is when our RNA polymerase, which is really just an enzyme that catalyzes this whole process, binds to a promoter and begins to unwind that double helix shape. That begins the process of an RNA molecule. Next comes elongation. That RNA polymerase begins to unzip the DNA that we already have, so that double helix shape starts to unwind. And in that process, the 4 bases that we talked about earlier, adenine, guanine, cytosine, and thymine, create the messenger RNA, that mRNA. Now, something to pay attention here is that thymine in this process becomes replaced by uracil. Quick change. Now, termination is kind of a misnomer because it's not really the end of anything, but the mRNA strand that will detach from that DNA sequence and travel outside of the nucleus, that's going to be really important for translation. That new mRNA strand has been made and ready to kind of start the process over or move on to translation. So that means we got to talk about what's next, which is translation. This is the process where the genetic information encoded in that new mRNA is now used to build a sequence of specific amino acids and therefore create a polypeptide. And we know that a polypeptide ultimately results in protein, and protein is what is responsible for life. Translation really is a series of events, and it begins what we just ended, where the mRNA molecule travels from the nucleus and ends up in a ribosome. Now, a ribosome can be found in a cytoplasm. In some cells, it can also be in the ER as well. I like to think of a ribosome as a protein-making machine. It has 2 halves to it, 2 subunits. The bottom half on your screen is nice, kind of a blue color. This is going to be the part that reads RNA. decodes it. The top half is going to be the subunit where it joins amino acids to form a peptide bond and ultimately a polypeptide bond. We'll take a look at that here right now. So when the mRNA has left the nucleus and has attached to a ribosome, as it's sitting there, the mRNA is going to be read by that part of the ribosome 3 units at a time. So on your screen right now, there is a green oval with a red triangle inside that says AAC. Okay, that would be 3 units at one time. You can also see on the screen there are 3 places where the word codon is being used. Codon is simply the word of the 3 units that are being read at a time. So on your screen there are 3 codon that are labeled. Now, at the same time, there is something called transfer RNA. And transfer RNA is going to be on the left side of your screen where it says anticodon. And anticodon matches up with the codon, but it also has a really important feature at the top. It has the amino acid connected. This is super critical for that polypeptide protein creation. So, those amino acids at the top all start to connect to one another as more mRNA molecules and anticodons make their way in. That's going to ultimately result in a peptide bond. And the more that happens and the more string of amino acids arrive, we finally arrive at something called a polypeptide bond. So the end of this sequence is really not the end of this sequence again, but this process will continue until a stop codon is received. In the screen in front of you, circled in red, that's going to be the sequence UGA. Now that's going to stop the mRNA sequence. It's going to stop the anticodon, the codon. It's going to stop the amino acid bonding. It's sort of the stop sign for this whole process, which leaves you with your final result. We finally have a polypeptide chain that's going to fold into a functional protein. Now, we have looked in class at these 4 stages that are on your screen. This arrival simply takes you to the primary stage. So from here on out, we've learned about secondary, tertiary, and quaternary. Okay, but transaction and translation get you to the first stage here. So here's what we've learned. We know that RNA is a copy of a DNA strand. Transcription is an RNA process of copying that original DNA sequence, making mRNA. Translation is the process of building a polypeptide chain with ribosomes and mRNA. And we result in protein, which is that polypeptide chain that folds in 4 stages to finally become functional protein. So where does that leave us? That's a lot of words and a lot of definitions. Where are we going from here? Well, as of April 2025, the federal government is petitioning for cuts to RNA research. And as we know, this research is critical. Now, according to the University of Edinburgh in 2019, mistakes during translation or in the creation of ribosomes cause a number of rare and incurable hereditary diseases, ranging from anemias to growth retardation syndromes. Now, other sources have suggested that somewhere in that process, you could also develop a probability or even diagnosis of cancer. To me, it's clear that RNA research cannot afford to be cut because this can simply just save lives or possibly even prevent diseases from spreading. We do know quite a bit about DNA and mRNA and all of the encompassing facts, but it seems to me that we just don't know everything or even enough. Thank you for staying, and I hope you learned some stuff. Great.